Being smart isn't one thing your genes control. It's at least 3 separate signals tangled together like overlapping radio stations. And scientists just built a way to tune each one in cleanly. What they heard on one particular channel wasn't what anybody expected. Psychologists have long known intelligence isn't a single dial. There's raw reaction speed, how fast your brain processes a simple choice. There's fluid reasoning, your ability to solve a brand new problem you've never seen before. And there's crystallized knowledge, the vocabulary and facts and know-how you've banked over a lifetime of learning. The catch is that these 3 signals overlap heavily in your genes, bleeding into each other like stations too close together on an old radio dial. Most genetic research studying mental illness just measured intelligence as one blurry composite, static, and signal mixed together, mainly because scientists never had a strong enough genetic data set to isolate crystallized knowledge on its own. A team led by Londono Correa and Elliot Tucker-Drob at the University of Texas at Austin wanted to actually separate the stations. Using genetic data pulled from roughly 439,000 people, mostly through the UK Biobank, the team first confirmed the 3 channels really were genetically distinct, not just 3 different names for the same thing. The genetic overlap between any 2 of the 3 channels came in well under half, meaning each one was carrying real, separate information. Only then did they build a more advanced statistical model that could isolate what's genetically unique to fluid reasoning and unique to crystallized knowledge, after filtering out everything the 2 channels share with each other, and with raw reaction speed. That filtering step matters because crystallized knowledge had never really had its own clean genetic signal before. It kept getting mixed in with the others. Once isolated, this dedicated genetic scan of crystallized knowledge turned up 78 distinct locations in the genome linked to it, published in Nature Communications in 2026. Several were completely new to cognitive science. One of the strangest, a handful of these locations were also tied to bone density, echoing an earlier finding where mice bred with a mutation in one specific gene linked to reasoning ability developed both cognitive problems and skeletal abnormalities at the same time. 2 seemingly unrelated body systems, apparently wired through some of the same genetic switches. And when the team checked which tissues in the body these genetic signals were most active in, both the fluid and crystallized channels lit up almost exclusively in brain tissue, frontal cortex, hippocampus, cerebellum, barely showing up anywhere else in the body, which is exactly the kind of result that tells researchers they're tracking something biologically real, not statistical noise. With clean signals for each channel, the team tuned in 5 neuropsychiatric conditions one at a time: schizophrenia, bipolar disorder, autism, ADHD, and Alzheimer's. Schizophrenia and bipolar disorder showed nearly identical patterns. Genetic risk for both was tied to modestly slower reaction speed and weaker fluid reasoning. But to stronger crystallized knowledge, and even to a separate genetic factor tied to staying in school longer, independent of raw smarts, with all 4 links reaching clear statistical significance. ADHD's genetic risk ran almost opposite on the first channel, tied to faster reaction speed, but was tied to weaker performance on every other channel, especially that school persistence factor, where the statistical odds of the link being random chance were smaller than 1 in a billion trillion. Autism's genetic risk showed no meaningful tie to reaction speed or fluid reasoning at all, but did show a real, positive tie specifically to crystallized knowledge, refining an older, cruder finding that autism's genetic risk correlates with cognitive ability in general by pinpointing which specific channel that link actually runs through. Alzheimer's risk stood apart from all of them, tied to only one channel, weaker fluid reasoning, and nothing else, matching how doctors already know memory and reasoning tests, rather than vocabulary tests, tend to catch early dementia first. Across 4 of the 5 conditions, one pattern kept repeating: weaker fluid reasoning. The solve it fresh channel tracked with higher psychiatric or neurodegenerative risk almost everywhere the researchers looked. Crystallized knowledge told a stranger story entirely. Here's the single clearest example of just how strange this gets at the molecular level. Buried in the genome is one specific variant, cataloged as RS1, 2, 4, 3, 3, 1, 0, 9. In this dataset, the version of that variant that nudges up your odds of schizophrenia is the exact same version that nudges up your crystallized knowledge score, measured independently in 2 completely separate groups of people. Not 2 separate genes doing 2 separate jobs. One switch, 2 effects, running in what looks like opposite directions depending on which outcome you're measuring. That's not an isolated fluke either. It's the same shape the broader statistical pattern takes across schizophrenia, bipolar disorder, and autism as a whole, just zoomed in on a single letter of genetic code. So why would evolution let genetic variants like that stick around if they raise psychiatric risk? The researchers point to a well-documented biological phenomenon called antagonistic pleiotropy, a single gene variant that costs you in one department while quietly paying out in another, so natural selection never fully weeds it out. Their working theory, backed by earlier published research they cite directly, is that some of these same variants also nudge up traits like curiosity, creativity, and openness to new experience. Traits independently shown in a separate, widely cited study to be predicted by the same genetic risk scores used for schizophrenia and bipolar disorder. A cost on one channel, a possible benefit on another. There's even a developmental clue pointing the same direction. Genes tied to crystallized knowledge showed their strongest activity in adolescence and adulthood, while genes tied to fluid reasoning were most active in early childhood and infancy. 2 channels running on completely different biological schedules, not just producing different outcomes. One honest caveat. This entire analysis draws only on people of European ancestry, and one of the crystallized knowledge gene hits turned out to be tied to skin tone. A reminder that some of these signals could reflect social and environmental patterns tangled up with genetics, not pure biology. The core pattern held up regardless. What holds up? Cleanly, across 4 separate conditions. Genetically weaker fluid reasoning consistently tracks with higher risk, making it the one channel that behaves the way common sense would predict. Crystallized knowledge refuses to play along, tangled up instead with traits that look from a different angle, like real cognitive and creative strengths. None of this says a good vocabulary makes someone mentally ill, or that struggling with logic puzzles protects you. These are population-level genetic patterns, not individual predictions. The researchers themselves flag a genuinely practical stake here. As genetic screening technology improves, selecting embryos or individuals for lower psychiatric risk on these particular gene variants could, without anyone intending it, also select against stronger crystallized knowledge, since in this data, the 2 are wired to the same switches. What it does say is that SMART was never one signal to begin with, and the stations we've spent a century treating as interchangeable turn out to be broadcasting genuinely different things about the mind.